Every human being alive today began with a single moment of release. One cell breaking free from the place it had waited in for decades, at exactly the right hour of exactly the right day. How does the body know which cell to release? And when? What actually causes it to break free? And why does this happen only once a month out of a supply that was decided before birth? Today, we're following that process from beginning to end, from a fluid-filled chamber deep inside the ovary to the moment an egg is caught and carried away. A woman is born with 1 to 2 million immature eggs already present in her ovaries. No new ones will ever be made. By puberty, that number has already fallen to around 300,000, and across an entire reproductive lifetime, only about 400 of them will ever be released. The rest are lost gradually, month after month, in a process of attrition that begins long before a single egg is ever needed. Each cycle, this attrition becomes visible. Hormonal signals called FSH and LH cause a group of roughly 15 to 30 follicles to begin developing at once, each one a small fluid-filled structure containing a single immature egg. Only one of these follicles, called the dominant follicle, will continue to full maturity. The rest gradually stall and break down. Inside the dominant follicle, the egg sits within a pocket of fluid called the antrum, surrounded by several distinct layers of cells. The layer closest to the egg is called the corona radiata. Around that is a cluster of cumulus cells, and beyond them, sheets of granulosa cells lining the follicle wall, producing the estrogen that drives the rest of the cycle forward. Outside all of this sit the theca cells, which respond to hormonal signals by converting other hormones into that same estrogen. Together, these layers form something closer to a small, pressurized chamber than a simple cell. An egg held in liquid, wrapped in protective tissue, and slowly building toward rupture over the course of roughly 2 weeks. As the follicle grows, its rising estrogen output signals the brain that maturation is nearly complete. Around the middle of the cycle, this triggers a sudden and dramatic spike in LH, known as the LH surge. Inside the follicle, this surge sets off a chemical cascade, CAMP, then KEG, then a wave of gene activity in the granulosa and theca cells. Almost immediately, these cells begin producing new signaling proteins, including EGF-like growth factors, along with a sharp rise in progesterone. Until this point, the egg itself has been held in a state of suspended development, frozen partway through cell division since before birth. The LH surge is what wakes it. Within hours, the egg completes its first meiotic division, shedding a small structure called a polar body, then pauses again, this time only one step away from being ready to meet a sperm cell. While the egg finishes maturing, the follicle wall itself is being taken apart. Granulosa and theca cells release a group of enzymes, including matrix metalloproteinases, that begin breaking down the collagen and connective tissue holding the follicle together. This is not a passive weakening. It is closer to active digestion, concentrated at a single point on the follicle surface known as the stigma. At the same time, blood vessels at that exact point begin to constrict, cutting off local blood flow and effectively focusing pressure toward one small area. In studies where this constriction is blocked, the follicle fails to rupture at all. Nature appears to clamp the follicle at the top like a drawn ring, ensuring it tears in one place rather than many. Once the wall has thinned enough at the stigma, a small opening forms. Cumulus cells at the surface emerge first, followed immediately by the egg itself, still surrounded by its cumulus mass. This is ovulation. In animal studies of similar processes, the rupture itself takes only minutes once the opening appears. The egg does not simply fall free. As ovulation approaches, the cumulus cells surrounding it secrete hyaluronic acid, causing the entire mass to swell into a soft, jelly-like cloud. This cushion holds the egg together as it passes through the rupture point and out of the ovary entirely. It does not drift for long. The fallopian tube ends in finger-like projections called fimbriae, lined with rapidly beating cilia. As ovulation nears, these fimbriae begin sweeping close to the surface of the ovary, and the beating of their cilia creates a current strong enough to draw the egg, cumulus and all, into the opening of the tube. Without this mechanism, the egg would have no guaranteed path forward at all, since it is released not directly into the tube, but into the open space surrounding it. Once inside, the tube cilia continue moving the egg forward, toward the region where fertilization typically takes place. Behind it, the ruptured follicle does not simply disappear. It collapses inward and transforms into a temporary structure called the corpus luteum, which begins producing large amounts of progesterone to prepare the uterine lining, in case this particular egg is fertilized. If fertilization does not occur, the corpus luteum breaks down within about 2 weeks, progesterone levels fall, and the cycle begins again from the start. Several beliefs about ovulation persist, and not all of them hold up. It's commonly assumed that the ovaries alternate, taking turns releasing an egg each month. Large studies have not supported this. Ovulation appears essentially random between the two sides, with roughly even odds either way, and no meaningful pattern of alternation from one cycle to the next. A related idea, that one ovary is generally more fertile than the other, also has no strong evidence behind it in typical cycles. Some women experience a brief, mild pain around ovulation, sometimes called Mittelschmerz. This is generally attributed to minor irritation in the pelvic cavity, and it has no established connection to fertility or egg quality. It's also worth noting that ovulation doesn't always land on day 14, as is often assumed. Timing varies from person to person and cycle to cycle, and in some cycles, ovulation doesn't occur at all. And while it's true that 2 eggs can occasionally be released in the same cycle, leading to fraternal twins, further releases after a pregnancy has already begun are exceptionally rare, since pregnancy hormones typically suppress any further ovulation. From the first stirrings of a dominant follicle to the moment an egg is swept into the fallopian tube, this entire process depends on a precise sequence of hormonal timing, cellular breakdown, and mechanical capture. Out of a supply decided before birth, only a handful of these cells will ever complete this journey. Every person alive today exists because one of them did. Thank you for watching. New hope, new life.